Welcome to Tax Solutions. Today we are going to have a wider view on bacterial cell wall. Now before starting on what is bacterial cell wall, let us have a quick recap on what is bacterial cell. So what is bacterial cell? A bacterial cell almost always are bounded by a chemically complex cell wall, and interior to this cell wall lies the plasma membrane. Its interior appears morphologically very simple, and the genetic material is localized, naked, and are not separated from cytoplasm. Now this is an typical structure of a bacterial cell. We can see that they are having a cell wall, and then we have a nucleoid that is naked, and few ribosomes are there all over the cells, and inside the cell wall we have this plasma membrane, and here lies the cytoplasm. Now next, move on to the bacterial cell envelope. Now the bacterial cell envelope mainly composed of three things: plasma membrane, cell wall, and the layers outside the cell wall, which we known as outer membrane. Now in the right hand side we can see a basic structure of cell envelope having its outer membrane, its peptidoglycan layer, its periplasm, and at the below we are having the inner membrane. Now let us move on to the bacterial cell wall. Now what is bacterial cell wall? So the bacterial cell wall is a layer which is fairly rigid and that lies outside the plasma membrane. It is most important for prokaryotic structure for several reasons, such as they give a shape to the cell, they provide the cell or they protect the cell from osmotic lysis and also from toxic substances. And the major component that is contained in a bacterial cell wall is known as peptidoglycan, or we can say it a murin. Now overview structure of a bacterial cell wall. Now before starting about the bacterial cell wall structure, let us say that the cell wall structure, according to cell wall structure, the bacterial cell has been divided into two groups, that is a gram positive, a gram negative. Now where does this gram positive or negative bacteria came from? Now a scientist known as Christian Gram, who developed a staining procedure which is known as a gram stain, it became evident to most of the bacteria. As we have discussed, the major component of cell wall is peptidoglycan. Now depending on this peptidoglycan, these two major group of bacteria is being differentiated. So gram positive bacteria, they have a thick peptidoglycan layer, and that is why they are stained purple color. Secondly, the gram negative bacteria, they are basically having a thin peptidoglycan layer and also having a outer membrane. That is the reason they are known as gram negative. Now moving on to peptidoglycan structure. Now peptidoglycan is an enormous mesh-like polymer that is composed of identical subunits. The polymer contains two sugar derivatives, that are N-acetyl glucosamine and N-acetyl muramic acid, which in short are known as NAM and NAG. Now they also have some amino acids such as lysine, diaminopimelic acid, glutamic acid, alanine, etc. Now if we see the amino acid of the peptidoglycan structure, we will find the most of the amino acid are in D-glutamic acid, because the presence of this D-amino acid protects against degradation of most peptides. Now the backbone of the peptidoglycan polymers is composed of alternating NAM and NAG residues. A peptide chain of 4 alternating DLN amino acid is connected to carboxyl group of NAM. Many bacteria replace mesodiaminopimelic acid with another diamino acid, which is usually a L-lysine. So in this structure you can see the peptidoglycan subunit compositions, that is composed of NAM and NAG. The NAG is N-acetyl glucosamine and NAM is N-acetyl muramic acid. NAG with lactic acid is attached to by an ethyl linkage. The tetrapeptide side chain is composed of alternating D and L amino acid. In order to make a strong mesh-like polymer, chains of linked peptidoglycan subunit must be joined by cross-link between the peptides. Often the carboxyl group of the terminal D-alanine is directly connected to the amino group of DAP. Now this is mostly found in gram-negative bacteria. As we can see in the first diagram, the D-ala is being connected to DAP, and in the next diagram we can see that there is a peptide linkage between D-ala to L-lysine. Between the bridge is known as peptide or peptide interbridge. This is for the gram-positive bacteria. Now, this cross-linking results in an enormous peptidoglycan sac that is actually one dense interconnected network. This sac has been isolated from gram-positive bacteria and are strong enough to retain their shape and integrity, yet they are relatively porous, elastic and somewhat stretchable. Now here in this diagram we can see the gram-positive and gram-negative bacteria, the cell wall structure between them, and in the next structure we can see the helical structure of the peptidoglycan layer. So the structural difference between the cell walls of gram-positive and gram-negative bacteria are responsible for the difference in gram-stain reaction. Now to know the structural difference, let us go through the gram-positive and gram-negative bacteria individually. Now let us move to gram-positive cell wall at first. Now gram-positive cell wall are normally having a cell wall that are thick and composed primarily of peptidoglycan. Peptidoglycan in gram-positive bacteria often contain a peptide interbridge. So as we have already discussed earlier, that is the peptidoglycan layer is quite thicker than that of gram-negative. We can see almost 90% of cell wall composed of peptidoglycan layer. Next, they also have teichoic acid, which is a negatively charged acid that helped gram-positive cell wall to maintain the cell envelope, protect from environmental substances, and they also help in binding to other host cells. Now some gram-positive bacteria have layers of proteins on the surface of peptidoglycan and also have lipoteichoic acids. Now let us move on to periplasmic space of gram-positive bacteria. Now what is periplasmic space or periplasm? Now the space lies between plasma membrane and cell wall is known as periplasmic space. Now in case of gram-positive bacteria, this space is quite smaller than that compared to gram-negative. Now in gram-positive bacteria, this periplasm has very few proteins. The enzymes that are secreted in gram-positive bacteria are known as exoenzymes, and they aid in degrading of large nutrients. Now moving on to a diagram composing of this gram-positive cell wall. So here we can see that is the periplasmic space between the plasma membrane and the cell wall. The cell wall is comprising of the peptidoglycan layer, we can see over here clearly, and outside the peptidoglycan layer we can see the teichoic acid and lipoteichoic acid, and this space of periplasm is too short and too small, we can say, and then after periplasm there is the plasma membrane layer. Now moving on to gram-negative cell wall. Now gram-negative cell wall are much more complex than gram-positive cell wall. They consist of a thin layer of peptidoglycan layer, almost 5 to 10% of the cell wall, and they composed of an outer membrane which is exactly outward to the cell wall that contains lipid, lipoproteins, lipopolysaccharide and etc. And one of the difference between gram-positive and gram-negative cell wall is that gram-negative cell walls do not possess any teichoic acids. Now, let us move on to the periplasmic space of the gram-negative bacteria. Now, the periplasmic space of gram-negative bacteria is quite different from the gram-positive bacteria. Now, in case of gram-positive bacteria, we have already seen that the layer of periplasm is quite shorter. In case of gram-negative bacteria, the periplasmic space is containing to amount almost 20 to 40% of the cell volume. Many enzymes are also present in periplasm. For example, the hydrolytic enzymes, the transport proteins and the other proteins. When the cell walls are disrupted carefully or removed without disturbing the underlying plasma membrane, periplasmic enzymes and other proteins are released and thus they are studied. Some of those periplasmic proteins participates in nutrient acquisition and other periplasmic proteins are involved in energy conservation. Other than that, some periplasmic proteins are also involved in peptidoglycan synthesis and in modification of toxic compounds that could harm the cell. Now here is a diagram of gram-negative cell wall. We can see clearly the peptidoglycan layer is much thinner than compared to gram-positive bacteria, whereas the periplasmic layer is much thicker than compared to gram-positive bacteria. We can see a thicker outer membrane also, which is much more thicker in compared to gram-positive bacteria and containing many proteins such as porine proteins, lipopolysaccharides, bronze lipoprotein, etc. etc. Now let us go to a brief detail on outer membrane of gram-negative bacteria. Now the outer membrane lies outside the thin peptidoglycan layer of a gram-positive bacteria. Now many lipoproteins such as bronze lipoproteins connect the outer membrane to the peptidoglycan layer. And other adhesion sites are also reported other than this bronze lipoprotein. Most of the cell wall composed of outer membrane, that is a lipopolysaccharide layer. Now here we can see the diagram of an outer membrane of gram-negative cell wall. Now over here we can see that the cell wall is being divided into two components, that is outer membrane and periplasm. And below the cell wall there is the cytoplasmic membrane, or we known as plasma membrane, and above the periplasm we are having the outer membrane, and between them we are having the periplasm associated with a thin layer of peptidoglycan structure. In gram-negative bacteria, as we have already discussed, that the peptidoglycan layer is much thinner, so it is attached to the periplasm. Other than that, in outer membrane we are having proteins like O polysaccharide, core polysaccharides, lipid A, porine proteins, lipopolysaccharides and etc. Now moving on to the lipopolysaccharides. Lipopolysaccharides are large complex molecules containing both lipid and carbohydrate, and they consist of mainly three parts, that is lipid A, core polysaccharides and the O side chain, popularly known as O antigen. Now LPS has many important functions. Firstly, it contributes a negative charge on the bacterial surface. As a major constituent to the exterior leaflet of outer membrane, lipid A helps to stabilize the outer membrane structure. LPS may contribute to bacterial attachment to surfaces during the biofilm formation. A major function of LPS is that it aids in creating a permeability barrier through restricting the entry of bile salts, antibiotics and other toxic substances that might kill or injure the bacterium. LPS also plays a role in protecting pathogenic gram-negative bacteria from host defects. The O side chain of LPS is also called the O antigen because it elicits an immune response. This response involves the production of antibiotics or antibodies that bind the strain-specific form of LPS that elicited the response. Now, although several explanations have been given for the gram-stain results, it seems likely that the difference between gram-positive and negative bacteria is due to their physical nature of the cell walls. Now, if the cell wall is removed from gram-positive bacteria, they stains gram-negative. Now, the peptidoglycan layer is itself not stained. Instead, it seems to add the permeability layer, preventing the loss of crystal violet. Now, during the procedure, the bacteria was first stained with the crystal violet and next treated to iodine to promote dye retention. When the gram-positive bacteria are treated with ethanol, the alcohol is thought to shrink the pores of the thick peptidoglycan layer. Thus, the di-iodine complex is retained during the short decolorization step and the bacteria remains purple. In contrast, the gram-negative peptidoglycan is very thin, not as cross-linked and larger pores. Alcohol treatment may extract enough lipid from the gram-negative outer membrane to increase its porosity further. For these reasons, alcohol more readily removes the purple crystal violet iodine complex and gram-negative bacteria, thus stained pink or red, which is the counter stain known as safranin. Now, this is the diagram of a gram staining procedure. Here we can see both the cells, positive and negative, both are at first not stained, and then they are being added crystal violet, which is the primary stain. Next, we add the iodine component to create a CVI complex, and then in this decolorization method, which is also known as the ethanol or alcohol step, which removes the crystal violet color from gram-negative cell. As a result, the gram-positive cells retains the crystal violet color, whereas the gram-negative cells, they are being secondarily stained with safranin, which in result give the pink or red color to the gram-negative cell. Now, we are going to learn about some components external to cell wall. Now, prokaryotes have a variety of structure outside the cell wall that can function in protection, attachment to objects and cell movement. Now, some of them are discussed. The outermost layer in the cell envelope sometimes consists of a network of polysaccharides extending from the surface of cell. It is referred to as glycocalyx, a term that can encompass both capsules and slime layers. Now, what are capsules? So, when a layer is well organized and not easily washed off, it is known as capsule. It is called a slime layer when it is a zone or diffuse unorganized material that is removed easily. So, from this definition, we can understand clearly that capsule is harder to remove, and when that is easier to remove, call it as a slime layer. Capsules are clearly visible in the light microscope when a negatively stains. Although capsules are not required for growth and reproduction in laboratory cultures, they do confer several advantages when prokaryotes grow in their normal habitat. They help in pathogenic bacteria resist phagocytosis by host phagocytes. They protect the cell from desiccation and also excludes virus and detergents. Slime layers are similar to capsules but diffused, unorganized and can be easily removed. Now, this is a diagram of capsule and slime layer. You can see the hurdle layer is known as capsule, whereas the layer which is diffused, we call it as slime layer. Now, many prokaryotes have a regularly structured layer known as S layer on their surface. In bacteria, the S layer is external to cell wall. In archaea, this S layer may be the only cell wall outside the plasma membrane. The S layer has a pattern something like float tiles and is composed of protein and glycoprotein. In gram-negative bacteria, the S layer adheres directly to the outer membrane. It is associated with the peptidoglycan surface in gram-positive bacteria. It may function in protecting the cell against the ion and pH fluctuations, osmotic stress, enzyme, and predation. The S layer also helps maintain the cell shape and envelope rigidity. It can promote cell adhesion to some surface. Finally, the S layer seems to protect some bacterial pathogens against host defense and contributing to their virulence. Now let us move on to cell wall and osmotic protection. Microbes have several mechanisms for responding to changes in osmotic pressure. This pressure arises when the concentration of solute inside the cell differ from that of outside, and the adaptive responses work to equalize the solute concentrations. However, in the certain situations, the osmotic pressure can exceed the cell's ability to adapt. In these cases, additional protection is provided by the cell wall. Now, for example, when the cell is in hypotonic solution, one in which the solute concentration is less than that of cytoplasm, water moves into the cell, causing it to swell. Without the cell wall, the pressure on the plasma membrane would become so great that it would disrupt it and the cell would burst, a process called lysis. Conversely, the hypertonic solutions water flow out and cytoplasm shrivels up, a process called plasmolysis. Here is the diagram of the three kinds of solution, that is the isotonic solution, hypertonic solution and hypertonic solution. So from here we can see an isotonic solution is there is no movement of water particles. In hypertonic, water particles moves out of the cell, whereas in hypotonic, water particles move into the cell. The protective nature of cell wall is most clearly demonstrated when the bacterial cells are treated with lysozyme or penicillin. The enzyme lysozyme attacks peptidoglycan by hydrolyzing the bond that connects the NAM and NAG. Penicillin works by a different mechanism. It inhibits peptidoglycan synthesis. Now, if they are gram-positive, treatment with lysozyme or penicillin result in the complete loss of cell wall and the cell becomes protoplast. When gram-negative bacteria are exposed to lysozyme or penicillin, the peptidoglycan layer is lost but the outer membrane remains. These cells are called the spheroplast. So now we are familiar with these two words, what is protoplast and what is spheroplast. Next we come to mycoplasma. So what is mycoplasma? Mycoplasma is the bacteria that lacks the cell wall. They do not have the ability to produce cell wall, but their plasma membrane are more resistant to osmotic pressure than those other bacteria having cell walls. So this is all for today. If you have any questions and any queries, do comment us to the comment section and let us know your feedback, and also let us know what else do you want from us. And if you like our video, please do subscribe our channel TAG Solutions. Thank you and have a nice day.